I'm a new postdoc at the Center for Evolutionary Hologenomics, and I've been here for about 6 months. So I won't give you too much of an overview on what hologenomics is, because I think most people in the section are actually in the center, and also Adam just gave a great overview. Thanks very much, Adam. So obviously, as an evolutionary biologist, and having focused so much in my PhD on how resource shifts contribute to local adaptation, and how I really wanted to understand how microbiomes can contribute to that as well, and also how microbiomes can shape or even drive these ecological processes that and evolutionary processes that I was just talking about. So my current project is embedded within the Earth Hologenome Initiative, which I also manage with Antton and a few other people now. So we just recently launched the Earth Hologenome Initiative, and we really believe it's different from other. It's not just a project, it really is a global endeavor coordinated at CEH, which has the objective of promoting, facilitating, coordinating, and standardizing hologenomic research on animal microbiota systems worldwide. So there's many different goals within the EHI other than just research goals. We really want to generate this paired animal genomic and microbial metagenomic data of wild organisms across the world. So we hope to get multiple individuals per population and multiple populations across climatic and different habitat gradients, so that we have the spatial host genomic and microbial metagenomic variation captured. And we can use this data for individual species questions, but also multi-species hologenomic analysis. A second goal of the Earth Hologenome Initiative is to create a network of interdisciplinary researchers contributing to the sampling of wild animals. So generating like a really large global data set like this only gets better the wider the net is cast. And we really want it to be as interdisciplinary as possible, because that will help us make the best decisions regarding the scientific questions we can answer, and also make sure we're doing it in the most standardized and appropriate way. So it really is based on collaboration, not competition in any way. And we already have over 100 researchers joined from across the world. Mostly it is pretty biased to the US, Canada, and Europe at the moment, but we also have some collaborators in Australia. And we really hope to reach the other continents as well. So just a brief overview of a few of our collaborators, where they're based. And the third goal of the Earth Hologenome Initiative is that sampling, preservation, and laboratory methods are all completely standardized to allow reproducible and comparable research based on open resources, so that everybody can contribute. And this also requires that standardized metadata on samples and the animals is also captured. So this is standardized, and sampling is really especially necessary for microbial data, as I'm sure a lot of you know, which is particularly sensitive to sampling and preservation methods to stop any community biases. So to facilitate this, we sent out a kit containing absolutely everything you would need to join the initiative, including tubes pre-filled with buffer and pre-labeled, as well as the swabs that you might need to collect any samples. So we can send these out to all the collaborators, and then we receive this kit back with everything we need to add to the biobank, which is the Earth Hologenome Initiative. And so last but not least, why we're all here is to do research. So a number of projects have already started to emerge from the Earth Hologenome Initiative. And some people have joined by just contributing individual samples, or some people have joined with already questions in mind and contributing samples with multiple species to answer maybe a larger question within that. So for example, we have individual populations across different climatic gradients, such as Bodarcis lizards. Different swallows are being contributed, such as house and tree swallows, and also falcons in Tenerife. We're getting temporal samples from them of them and their chicks to answer individual questions, but then these questions or these samples also get added to the database to answer multi-species questions. So we also have are getting marsupial samples from Australia and species that are their placental counterparts in similar ecological niches, to compare and try and understand how placentals in Australia have evolved to the novel ecological niche. So with this, we can answer so many questions. Like really the possibilities are endless depending on people and other people joining and collaborating with us. So just like very, very broadly and not limited, we can answer questions such as speciation, adaptation, invasion, hologenomics, and conservation. So just to go into like a bit more depth on one project that has emerged from the EHI, and which I think my postdoc will be primarily based on, is Squirrel Hologenomics, which already has a logo. And the aim is to study the correlations between the ecological niche properties of the species of both red and grey squirrels across their global distribution. So across different environmental gradients, such as climatic gradient. And also we're doing more local studies, hopefully, where there's already one taking place in Italy. And we're hoping to get more local scale studies where the species exist as isolated populations from each other, but also they exist in symmetry, to look at, also look at urbanization gradients within the species and how they adapt to a more urban environment. So like all data that goes into this, we can ask specific questions with other species, or also it all gets added to this multi-species database tab, so we can answer questions that entail the analysis of multiple species. So just to go into a bit more detail on the sampling, because just to show you how we actually aim to achieve this and the methods that we're going to be using, and give you all the information, because I'm sure so many people out there are sampling right now, or with COVID, probably not, but in the future plan to sample wild animals and would love to get involved. So just to begin, for comparative analysis, we've come up with this like hierarchical design to facilitate as many people being able to contribute samples as possible. So our very first requirement is that we get samples that are feces and or cloacal anal swabs. This is dependent on the species, obviously, of what you can get, to capture the microbial community of the lower intestine. And there'll also be a little bit of host DNA in there, but probably not that much. And just also all samples are captured from all samples are obtained from live captured individuals. So the appropriateness of the sample really is dependent on the species and what you can get as well. So on top of feces, we ask for oral swabs potentially, for which you can get for some species, but not others, because that will help us get not only the oral microbial community, but also get more host DNA to get the full hologenomic picture. And so if you can't get oral swabs, another option, depending on the species, is tissue or blood samples. So tissue samples if you work on maybe mammals, but blood samples if you work on birds, because there's more DNA there. And that will help us get not only just the gut microbiome, but the host DNA. Okay. So just a fourth thing to keep in mind, if anybody here works on amphibians, it's also great to get skin swabs, since we know that the skin microbiome of amphibians really contributes to adaptation and acclimation. So, and then just to give an overview again, this hierarchical design will really facilitate as many collaborators as possible, but also consider the limitations a lot of researchers will have on just even sampling permits. Like, well, somebody might not be able to get tissue samples from squirrels in one country, and others, the red squirrels, might not be able to get an oral swab because they're just like what Adam's been dealing with, aggressive maybe. It's hard to guess. So this will just allow us to get both all the hologenomic data we need, and everything to be as comparable as possible and reproducible. And once samples are given to EHI, we store them, and all the samples follow the same standardized laboratory and bioinformatic procedures. So that is something that has to be very strict based on the fact that, like I've discussed, microbial communities are very subject to the communities can become quite biased depending on different procedures. But we really hope that despite that, other people not only just collaborate by contributing samples to the initiative, but also contribute to the analysis. And just as an example, we already have, like I said, over 100 collaborators out there and maybe 20 kits sent out to people. It's been a slow start because COVID. But we really hope that 2020, it's already starting to ramp up very quickly. And we have a lot of people out sampling first at the moment, and for a variety of different reasons. So some people have been sampling for educational purposes, and then they were just reached out to us because they thought, why not just actually get some samples in a standardized way and make this for another reason other than just educational purposes. So Ange Charmantier down there is, I want to say, sampling a tit in France. And then other people are sampling and accidentally or capture non-target species. So they're able to get a wide variety of species that they're not actually even interested in. And it's really not that much extra work to just take another couple samples from other individuals to contribute to the Earth Hologenome Initiative, such as a little field vole down there has been caught in a trap. And other people then, like I've said, contribute to with actual research questions in mind and can contribute all the samples regarded for, or all the samples just for one individual project already. And then that will all go into this multi-species larger scale questions. So really, this whole talk is just basically to show how you can get involved if you want. And a project like this really is there's so many ways, and we really hope that as many people can get involved as possible. The potential scope is just really large. Even if you're a hobbyist, I don't know about here, but at home, people are out mist netting and ringing birds just for the hell of it. So I wouldn't be shocked if that was the same here.